first, I just want to thank Dr. Ali for a really, and Dr. Samira for a really informative talks that lead really nicely into what I'm going to speak about right now. I think it was really great that Dr. Ali's talk really laid the groundwork for what the scene is with respect to malaria vectors. And in my talk, I'm going to focus much more on Aedes aegypti and the monitoring and management of insecticide resistance in Aedes aegypti. So first, just why is insecticide resistance so important, especially with respect to Aedes aegypti? Well, there's a limited number of insecticides approved for public health use, which you all know very well. The four principal classes are listed here, although I should say, especially for malaria control right now, there are a couple new classes that are pre-qualified, but we are still quite limited for Aedes aegypti. And then, of course, there are no vaccines or chemotherapeutics for chikungunya and Zika. And the vaccine that is available for dengue is quite limited and not licensed in many places. So given this scenario, effective mosquito control remains our principal strategy for preventing and controlling most Aedes-borne diseases. So for insecticide resistance monitoring, the most important thing to bear in mind is that this should occur in a routine and systematic fashion and as a key component of vector surveillance and control programs. Susceptibility should be assessed to the active ingredients available in the insecticide-based products that are approved for public health use in whatever country is conducting the surveillance. So really, whatever products that are available to your programs, the insecticides in those products are what the priority target should be for monitoring insecticides in the country. And in the case of Aedes aegypti, there's quite a bit of larval control as well. So in that sense, if larval control is also being conducted, resistance should also be monitored in both larval and adult mosquitoes. And then the third point I have here is that the monitoring should occur at operationally relevant scales. And I'll touch a bit more on this in a few minutes because the spatial variations in insecticide resistance can be even more extreme in Aedes aegypti than what we see with Anopheles. So how do we test for insecticide resistance? And Dr. Samira and Dr. Ali already alluded to this and spoke a bit about it. But the first step is to conduct bioassays. Now, it's really important to understand what a bioassay can tell us and what a bioassay doesn't tell us. So what bioassays do is they serve as an early warning tool to detect changes in the ability of a discriminating dose of an insecticide to kill or incapacitate mosquitoes in a given population. So if we're looking at a population over time and conducting bioassays once a year, a couple times a year on the same population, if we start to detect changes in the amount of mosquitoes from that population that are dying, that's our early warning that that population is developing resistance to that particular insecticide. So I've put in red on this slide that changes in susceptibility that we detect via bioassay don't necessarily translate into operational failure. So say you're studying a population, you're monitoring it, and you start to detect some decreased susceptibility to that insecticide. It doesn't mean that any intervention that's using that insecticide right now is going to fail, but it's an early warning sign that your population is starting to develop resistance. So you should go ahead and switch your product before the resistance gets to a point where it actually does result in operational failure. So it is very important to understand that when your bioassays tell you you have resistance, it's not automatic that your control is failing, but it is certainly an indication that you need to shift to something different. So for the bioassay methodologies, there are two principal ones. Samira mentioned the WHO bioassay, and there's also this CDC bottle bioassay. And both of these will be part of the e-practical on Thursday. So we'll get into a lot more detail then about how these are run and the specifics of it. But just to very briefly say that the bottle assay, the CDC bottle assay, looks at what's happening to the mosquitoes at a particular that are exposed to a particular dose of an insecticide at a particular time. So for instance, if we had an 80s population that we exposed to bottles treated with bendiacarb at the diagnostic dose, which is 12.5 micrograms per bottle, we look at the time at 30 minutes, what proportion of those mosquitoes are still alive and what proportion are dead. So instead of waiting for the 24-hour holding period as one would do in a WHO assay, this is a much more acute time dose response assay. And again, we can get into the specifics of this and talk a bit about this methodology versus the WHO papers a bit more on Thursday. But suffice to say, the thresholds are the same with the CDC bottle assay. If 98 to 100% of your mosquitoes are knocked down or dead at the diagnostic time, you can consider that population susceptible. If it's between 90 to 97%, that's a sign that the population may be developing resistance. And if less than 90% are knocked down or killed, that population is considered resistant. And this is just a picture showing the WHO cylinders that use the treated papers, which I'm sure most of you are familiar with already. And again, the details of how that test is run will be elaborated on Thursday. So there's quite a lot of data out there on phenotypic resistance in Aedes, bioassay data that show where resistant populations of Aedes are. And I just wanted to highlight this excellent paper that came out several years ago that at that time attempted to collate all of the data that were available on Aedes resistance and show what it looked like across the world. And this is just one of the images from that paper showing deltamethrin resistance over the period between 2006 and 2015. And you can see that there's a lot of gaps on this map. There's a lot of resistance monitoring that happens in South America and in Southeast Asia, particularly, Thailand, Malaysia, and Singapore. But when we look up, in the Middle East, North Africa region, there's not a lot of data, or at least there weren't that were included in this particular study. And a lot of that could also be due to the fact that this study looked largely at what was available in published literature, and national programs are probably doing more monitoring than what this captured. But what it shows is that there's quite a lot of resistance that's been reported in the Americas and in Southeast Asia. But there are a lot of unknowns on this map. But the fact that there is a lot of resistance is troubling and is a sign that vigilance should be increased across the world. So bioassays are really the first step just to get an understanding of if a population is developing resistance to an insecticide. But once a program has a routine way to monitor populations and there is resistance that's being detected, it's then also important to try to characterize what some of the underlying mechanisms of that resistance are. And Dr. Ali already touched on a lot of this in his talk, but I'll try to give it a bit more of an 80s specific spin to it on this one. So there are four, I guess, main mechanisms of resistance. And the two that are most commonly studied and that we know the most about are the first two that are listed here. Number one being target site insensitivity. And this is when point mutations prevent the binding of an insecticide at the target site within the mosquito. And the two most common target site mechanisms that are most commonly studied, at least in mosquitoes, tend to be KDR and insensitive acetylcholinesterase, or the ACE1 gene. So the second one that's listed here is metabolic resistance or increased detoxification. And this is what happens when there's heightened enzyme activity in the mosquito that leads to increased detoxification of insecticides. And this is something that is also decently studied now in Aedes aegypti. We're still figuring out a lot of it, but through biochemical assays and RNA types of gene expression assays, we're learning more and more how metabolic resistance works in mosquitoes. Now, the third is a really up and coming area of research, and that's cuticular resistance. And this is what happens when changes in the mosquito cuticle prevent the penetration of insecticide, usually due to thickening of the cuticle. This has not been something that has been well studied until recently, and it is quite troubling to think that if a mosquito cuticle becomes sufficiently impenetrable or thick, it could cause cross resistance across most insecticides. So this is one that is a very serious concern. And then the fourth one is behavior changes. And this is, there's anecdotal evidence that shows this happening. There's not a lot of research in this area per se, but there's two ways to think about behavioral resistance. One is when a species of mosquito adapts their behavior so that they're less likely to come into contact with an insecticide-based intervention. And I think the best example of this are some populations of Anopheles gambiae in Africa, whereby they used to bite inside houses at nighttime. So when people were sleeping under an insecticide-treated net, the mosquitoes would come in. They wouldn't be able to get to the host that they were looking for. They would take up a sufficient dose of insecticide and die. But what we've seen is there's been shifting of behavioral patterns so that mosquitoes are now in some places biting not so late at night, maybe not so much inside houses. So on a population level, the behavior has adjusted or shifted due to the type of insecticide-based interventions that have selected for that behavior. So that's one way to look at behavioral resistance. But another is really basic mosquitoes not responding to volatile insecticides. So for instance, some insecticides are highly volatile. It means they are living, they're in the airspace rather than stuck to a surface. And in that instance, you can have mosquitoes that don't really sense the insecticide until it's too late and they've come into contact with a lethal dose in the air and they die. But then there are ones that respond to that stimuli. They can sense that, oh, there's some volatile insecticides there. I'm going to move away, and I'm not going to go into that area where that volatile insecticide is. So that's another way that mosquitoes can avoid coming into contact with insecticides, and that's a type of resistance that they can develop. So all that to say, these are the four kind of principal mechanisms. And while these mechanisms may exist alone, they more commonly exist in combination with each other. So you'll have a mosquito population that is probably going to have some level of multiple types of mechanisms underpinning their resistance. So this is just an alternate way of looking at that really nice graphic that Dr. Ali showed about how different mechanisms can be shared across the four major insecticide classes. So not to belabor the point, but there's target site insensitivity that can cross between classes, and there's different enzyme groups that can also cross between insecticide classes. So that's just something important to bear in mind if we think we're going to switch from a carbamate to an organophosphate. We really need to have a good understanding of what's causing the resistance to perhaps the carbamate, because if it's primarily esterase based and it's the same esterases that are causing resistance to organophosphates, it might not be wise to select a particular organophosphate. So it's really quite a complex web of things that are happening that cause resistance in mosquitoes. But I guess there's a bit of good news in that too, in that what we're seeing with all of this increased study into this area is that resistance to one insecticide within a class does not necessarily mean that a population is resistant to all insecticides within that class. So I don't think we're quite at the point of, recommending within class rotations as part of insecticide resistance management, but in settings where there's limited tools available for use, it just offers us a bit more flexibility. So I'm just going to speak a bit about some of the more specific mechanisms of resistance and what we know in Aedes aegypti. So with respect to target site resistance, as I mentioned, there are two really key ones that we know about. The first being KDR, and this is caused by point mutations on the voltage-gated sodium channel gene, and it can result in resistance to DDT and some pyrethroids. Now, in Aedes aegypti, it's interesting because there are multiple mutations that have been reported, some of which are associated with phenotypic resistance and some not. So you can actually have, populations that are susceptible, and you can still be picking up some of these alleles. So it's important to understand which combinations of KDR alleles are really the ones that contribute the most to resistance that's seen in a mosquito population. Now, with respect to ACE1, the insensitive acetylcholinesterase, the ACE1 gene, at least in Anopheles, there are point mutations on that gene that can result in resistance to organophosphates and carbamates. And while it's been pretty commonly reported in Anopheles species, it has only recently been detected in Aedes in Asia, and it's not really recognized as a widely important mechanism in Aedes, at least not yet. So this is just a nice representation of where the different KDR alleles had been detected in different continents. This comes from that Moya's paper, which I showed earlier. And I mean, I think we can probably populate this a bit more now because there has been more research in the few years since that paper came out. But just to show that there are multiple mutations multiple KDR mutations, some that are associated with resistance, some that are really only associated with resistance when it found in combination with some of the other mutations. But then there are others that so far there hasn't really been an association with resistance that's been demonstrated. So there's a lot to look at here, and it's not just a question of reporting KDR frequencies and hey, then we'll know if our population is resistant or not. Unfortunately, it's not that simple. It'd be great if it was that simple, but it's not. It's a bit more complex than that. And again, this paper predated the finding of the 410 mutation, which actually has been shown to be quite important in Aedes aegypti, KDR being associated with phenotypic resistance. So then what does KDR actually tell us about resistance in Aedes aegypti? Everybody likes to look for it and report it, but what does it all mean? So what we can say is that the 1534C mutation arose first and is more directly associated with resistance to permethrin and DDT. Then subsequently, the 1016, either I or G, and the 410 leucine arose. And these are more associated with pyrethroid resistance, but mostly in combination with 1534C, simply because this one came first, 1534C, and on that background, these others came along as well. I think that if we think about it, KDR, what it does is it provides us with a nice marker that demonstrates that the population is under selective pressure. As I said earlier, we can't really say that, oh, well, if I've got this KDR mutation at this frequency, you know, my population's going to be resistant to deltamethrin. Not really. You still got to do the bioassays, and there's a lot of other things that can be causing that resistance. But what the KDR, but monitoring KDR does give us a quantitative measure of selection pressure in a given population. So it can be quite useful in that sense. And one thing that's quite unclear in Anopheles, again, KDR can confer resistance, but the most intense resistance in Anopheles is usually due to metabolic mechanisms or other factors. And in Aedes, it's still not really clear yet how this all shakes out. But if it is similar in that respect, really the metabolic resistance in Aedes is probably going to be what causes the really intense resistance that's out there. So let's talk about metabolic resistance. So metabolic mechanisms of resistance result from usually from the overproduction of detoxification enzymes that then metabolize xenobiotics, including insecticides. So it just makes the mosquito much more efficient at breaking down that poison before the poison has a chance to kill the mosquito. So in the case of pyrethroids, the most commonly implicated enzymes are the cytochrome P450 oxidases. However, in Aedes aegypti, we're actually seeing that the glutathione S transferases may be particularly important, and that's interesting, and that's something that there's still not a lot known about, but there do seem to be some trends in that direction, more so than in Anopheles. So this overproduction of enzymes is typically due to the overexpression of genes that code for those enzymes in the mosquito. So we can do gene expression studies to look to see relative to a susceptible strain or a housekeeping gene, whether certain genes that code for enzymes are being overexpressed. and that can lead to the over production of those enzymes. But then there's also mutations, single nucleotide polymorphisms on some of these detoxification genes that have also been associated, at least with delta methrin resistance in Aedes aegypti. So really getting DNA markers of gene enzyme overproduction in mosquitoes is something that is not quite as well studied yet in Aedes aegypti as it is in Anopheles, but there are signs that there are actually some SNPs out there that are associated with this type of resistance. So how do we detect metabolic mechanisms of resistance? Um, well, there's three primary ways that it is done. The first is simply through synergist assays. And this is when we pre-expose mosquitoes to a synergist before conducting our typical insecticide bioassay. And depending on the synergist we use, we can estimate the degree to which the resistance that we're observing is conferred by either oxidases, esterases, or GSTs. So it's on sort of a crude level. We can get an understanding of how much susceptibility can be recovered in a population if we expose those mosquitoes to one of these synergists before exposing them to the insecticide. The next sort of way to look at this is through biochemical assays. And those were some of the pictures in Dr. Ali's presentation that were showing how those biochemical assays are conducted. And what these assays do is they measure the level of activity of some of these large enzyme families compared to insecticide susceptible control mosquito strains. And then finally, there are molecular assays, which are typically RNA based, at least for Aedes aegypti at this point. And these are meant to detect differences in the levels of gene expression for genes that then metabolize specific insecticides. So this is just a quick schematic of how synergist bioassays work. And I think we can talk about this in greater detail on Thursday during the e-practical. But really, what you would do is first expose the mosquitoes to a certain dose of a synergist, before then letting them uh, uh, rest for a bit in the holding cartons, and then putting them into the bottles or the WHO cylinders that are treated with the insecticide. So ideally, you would do that in parallel, like with a typical bioassay. So you would know how much resistance you have in your population without exposing it to a synergist first, and then how much resistance is still there, if any, when you've pre-exposed that population to a synergist. So for biochemical assays, these require fresh or frozen non-blood fed mosquitoes. Just because blood fed mosquitoes, there's a lot of enzymatic activity going on. The metabolism is already altered in the mosquito because of digesting the blood meal and other things that are happening. So in the instance that you just collect fresh mosquitoes from the field, the abdomens should be removed, and then the assay is just done on the heads and thoraces. So you can detect levels of, again, the large enzyme families, oxidases, esterases, GSTs, and you can also look for the inhibition of acetylcholinesterase, insensitivity in acetylcholinesterase. And then there's a Bradford assay that's used to measure the total protein content of each mosquito. And this just allows for corrections due to just biological variations in mosquito body sizes that could exist. So again, Dr. Ali went through this already. The mosquitoes are analyzed in triplicate in a microplate well, and it's a colorimetric test. So you can often just read it by eye, but you can also use a plate reader to get really precise measurements to determine what degree of activity you have with respect to certain enzymes. And this is just an example of what those data would look like. These are just different populations of Aedes aegypti from Colombia. And you can see there's quite a bit of variation between activity levels of oxidases, esterases, and GSTs with respect to the Rockefeller susceptible strain, which was the control strain that was used. So the genetics of metabolic resistance in Aedes aegypti is a really up and coming area of research. And there's a lot that's being looked at here as we try to tease apart what is really happening with the specificity of different mechanisms. So this just summarizes some Aedes populations from different parts of the world and genes that have been detected as overexpressed in these populations. And what I want to draw your attention to is just the variety. I mean, there are some, typical culprits that you see popping up with a certain degree of frequency, but there's a lot of variability between populations. And understanding how that variability is actually contributing to the phenotypic resistance we see is something that's a very important area of research. And again, this is like the KDR graphic that just summarizes what some of these specific metabolic mechanisms are and how they actually relate to resistance to different types of insecticides and in different parts of the world. Again, this is from that Moyes paper, and I would really encourage you to have a look at that because even though it's a few years old, it really does offer a very comprehensive overview of Aedes aegypti resistance.
Audrey, the time is nearly up, but we would very much like to hear more from you. Certainly, we'll have enough time on Thursday, but you still have a couple of more minutes to finalize. Is
Okay,
that all
I
right?
think I'm getting to the end, so I'll speed it up a little bit.
Sorry for the interruption.
No problem. Yeah, I am just finishing here. So I just wanted to say that in addition to those, there are other potential mechanisms of resistance that are cropping up and people are looking at. And this is just a paper that came out a couple years ago where we found some proteins that interact with the sodium channel are also genetically associated with pyrethroid resistance. And there might be some structural reasons behind that are inhibiting the binding of the insecticide. So some kind of atypical mechanisms might be out there. And then RNA sequencing, and this is something that our lab at CDC is quite heavily involved in at the moment, where we're finalizing some pretty comprehensive analyses from Mexico and Puerto Rico. And even in the RNA sequencing data, we're getting some hits for cuticular genes, which is a little bit troubling, as I mentioned earlier. So molecular assays to detect resistance mechanisms. I think Professor Lee is probably going to go into this in greater detail. This is just a real-time PCR that is used often to look for KDR alleles in Aedes aegypti. And to close, I just wanted to highlight some key challenges for resistance management. One is the fine-scale spatial heterogeneity of resistance patterns in Aedes. These can vary a lot over very small geographical areas. There's multiple sources of insecticide selection pressure on Aedes populations. People buy household sprays and insecticides use that around their homes, and that's an additional source of insecticide pressure that's not coming from national control programs. And ultimately, it remains unclear the degree to which insecticide resistance is compromising Aedes control and how that then translates into increased epidemiological risk of getting any of the Aedes-borne arboviruses. So just a few papers here that really describe field examples of this really fine scale heterogeneity in insecticide resistance. And I think you all have access to this presentation on the shared drive, and you can hopefully see these papers later. And then that selective pressures aren't only coming from control programs, and there's increasing evidence that household insecticide use can really maintain and even drive insecticide resistance patterns in Aedes aegypti. So how can resistance be managed? Well, as Dr. Samira showed, there's this very nice global plan for insecticide resistance management in malaria vectors. So it's focused on malaria vectors, and most of the examples that it proposes are hypothetical and logical and arise from experiences in agriculture. But there's not a lot of empirical evidence that's available for what works in malaria vectors. So what about Aedes? How can Aedes resistance be managed? Just like Anopheles, there's very little empirical data on how resistance can be managed in Aedes. And we don't really know the extent to which the recommendations of the GPERM also apply to Aedes. Again, they're logical. They've worked in agriculture, but how do you actually apply them in an operational setting where your focus is on Aedes mosquitoes? And then also, I think especially with Aedes, the environmental management and non-insecticide-based control methods are even more available and should be included as part of integrated vector management strategies. So there was a paper that came out a few years ago that proposed a framework for Aedes resistance management. And I'll let you come back and look at this on your own time because there's a lot happening in this. But suffice to say that the real cornerstone of this is knowing what's happening through bioassays and understanding the mechanisms of resistance. And all that feeds into being able to inform vector control decision making and monitor the efficacy of what's happening in the field and how resistance tracks with that. So to conclude, just to emphasize that resistance monitoring is a critical component of vector surveillance and control programs, and that data arising from resistance monitoring should be used then to inform vector control decisions. And that understanding the mechanisms of resistance can be really important and informative to determine the most suitable strategies for insecticide resistance management. So with that, I'd like to thank you for your attention and my apologies for going a bit over.